Today we're continuing to talk about passive transport so we can understand how chemicals move across the cell membrane, and specifically we're going to deal with facilitated diffusion today. So water, oxygen, carbon dioxide, these are all small molecules, which means they can pass through the cell membrane easily. They can slip between the phospholipids to get to the other side. But larger molecules like glucose, C6H12O6, are much too big to pass through the phospholipid bilayer. Similarly, charged particles, ions, because they have a full charge, they are too charged to get through. The actual charge will repel them out of the phospholipid bilayer if they try to get through. So in order to diffuse through the cell membrane, you have to be uncharged and you have to be small. But we need charged ions inside the cell, and we need those large macromolecules, which literally just means big molecule, in the cell as well. So what do? Well, what the cell does is it does something called facilitated diffusion. Facilitated diffusion uses transport proteins embedded in the cell membrane to help get these chemicals through. And what these transport proteins do is they basically provide an alternate channel so that your macromolecules and your ions don't really have to interact with the cell membrane directly. They can use the transport protein instead. Now, this still works off of the principle of diffusion, so moving from an area of high concentration to an area of low concentration, so it's still a form of passive transport. It doesn't require the usage of any energy. But we have the proteins there just to make sure it can take place, which is why it's facilitated diffusion. The proteins are facilitating the process. Now, each of these proteins has a specific molecule that it helps transport, and it identifies these by shape, size, and charge, same as the rest of the membrane does. And these function off of what we call the lock and key model. And this is an analogy between the protein and the chemical it's transporting to a lock and a key. There's only one type of key that fits in a particular lock and can turn it. Depending on how specific you are with your keys and how many you give out, there might just be one key. So there's only one type of molecule that can actually fit inside the protein in order to get through. Just like the one key turns the lock, only one chemical will be able to fit in any given protein. So the proteins distinguish between different types or different species of molecules and ions. We have two types of transport proteins as well. We have carrier proteins and we have channel proteins. They both work the same way. They both are facilitated diffusion, so passive transport, no energy usage, and they both have a specific shape that only allows a particular type of molecule or ion through, so they work on the lock and key model. The channel proteins get used more often with small ions, and they're just a literal water-filled passage that the ions can flow through. So all it's doing is it's just insulating the ion from the cell membrane so that the ion can get through. And these are actually specific to types of ions as well. So it's not based off of just size. The protein can actually tell what type of ion it is and keep the other ones out. The carrier proteins, on the other hand, change shape to allow things through. Now, this shape changing doesn't require energy, unlike the active transport we will see later. It's just that when the particular molecule that protein transports binds onto it, it causes the carrier protein to change shape. There's no requirement for energy to be inputted. This is an automatic process that doesn't take energy, and then when it unbinds on the inside, it changes shape back to its original. And these are what the two look like. So carrier proteins are on the left. You can see the before and after, that once it binds in, it changes shape, and that lets the molecule through. On the other hand, here we have a channel protein. And what the channel protein is doing is it's allowing ions to pass through the middle of the channel. There's no opening and closing. It's just insulating them from the cell membrane so they're able to get through.